Every cell in your body, with a handful of exceptions, contains mitochondria. These are not simply the powerhouses of the cell, a phrase that has been repeated so many times it has stopped meaning anything. They are electrochemical machines, specifically proton gradient-driven electron relay-powered ATP synthesis machines. And to understand what happens inside one, you need to follow a single electron from the food on your plate to the water molecule it eventually becomes. The inner mitochondrial membrane is almost completely impermeable to ions. That is not a bug, that is the entire design. Because it means that when proteins embedded in this membrane pump positively charged protons from the inside to the outside, from the matrix to the intermembrane space, those protons have nowhere to go. They pile up. They generate a charge difference, a voltage, approximately 220 millivolts across a membrane 8 nanometers thick, which, if you scale that to a macroscopic thickness, is equivalent to about 30 million volts per meter. This is one of the most intense electrochemical gradients in nature, and it is the direct source of the energy your cells use to make ATP. The proton pumping machinery in the inner membrane needs fuel. That fuel arrives as electrons, carried on two molecules generated by the Krebs cycle, the metabolic loop running continuously in the mitochondrial matrix. The first is NADH. Each turn of the Krebs cycle generates 3 NADH molecules. NAD+, the oxidized form, picks up a hydride ion from metabolic intermediates. NADH carries those electrons to complex 1 of the electron transport chain. Its reduction potential, negative 0.32 volts. It is, in electrochemical terms, highly motivated to give electrons away. The second carrier is FADH2. This one has a more specific origin. The enzyme that converts succinate to fumarate in the Krebs cycle is itself embedded in the inner membrane, and is, in fact, complex 2 of the electron transport chain. It never releases its electrons as a diffusing molecule. It hands them directly inward. This distinction matters. NADH feeds complex 1, which pumps 4 protons. FADH2 feeds complex 2, which pumps none. Different entry points, different ATP yields, same downstream destination. Complex 1, NADH dehydrogenase, is the largest protein complex in the chain. 45 subunits, an L shape, one arm in the membrane, one arm reaching into the matrix. NADH docks at the matrix arm's tip, hands 2 electrons to a flavin mononucleotide cofactor, and those electrons travel along 8 iron-sulfur clusters like beads on a wire. The wire terminates at ubiquinone in the membrane. 4 protons are pumped. The electron transfer is coupled to a conformational change, a physical flexing of protein structure that works like a molecular piston. Complex 2 adds electrons from FADH2 into the same ubiquinone pool. No proton pumping. 4 subunits, 3 iron-sulfur clusters. Electrons relay from FAD through the FES chain to ubiquinone. Ubiquinone, coenzyme Q, is the ferry, a small, fat-soluble quinone molecule that diffuses freely through the lipid bilayer, collecting electron pairs from both complex 1 and complex 2. Complex 3, the cytochrome BC1 complex, faces a fundamental mismatch. Ubiquinol delivers 2 electrons, but cytochrome C, the next carrier, accepts only 1 at a time. The solution is the Q cycle. 1 electron from ubiquinol goes forward to cytochrome C. The second electron travels backward through cytochrome B to reduce a new ubiquinone. Net result, 2 electrons reach cytochrome C, 4 protons are pumped. Cytochrome C carries each electron individually to complex 4, cytochrome C oxidase. This is the terminal enzyme, 13 subunits, 2 heme groups, 3 copper ions. 4 cytochrome C molecules each deliver 1 electron. 4 electrons reduce 1 molecule of oxygen to 2 molecules of water. 4 protons are consumed from the matrix. 2 more are pumped across. 10 protons per NADH pair. All pumped into the intermembrane space, building a gradient of concentration and charge, the proton motive force, approximately 220 millivolts. The only way those protons can return to the matrix is through ATP synthase, complex 5. ATP synthase is a rotary motor. Protons flow through its F0 subunit, rotating a central shaft at 100 revolutions per second. That rotation sequentially compresses the 3 catalytic sites of the F1 head, each time forcing ADP and inorganic phosphate together into ATP. 4 protons, 1 ATP. Repeat continuously for as long as you are alive. Now consider a molecule that is flat, planar. 3 aromatic rings fused into a single conjugated system. 2 benzene rings flanking a central ring containing both a sulfur atom and a positively charged nitrogen. The entire backbone is one continuous pi electron cloud. Because that cloud is so extended, the molecule can accept an electron without undergoing structural disruption. The energy is spread across the whole system. It is extraordinarily easy to reduce. And because of the electron-donating dimethylamino groups, it is also eager to give those electrons away once reduced. It fills, it empties, it fills again. Its reduction potential, positive 0.01 volts. NADH sits at negative 0.32, cytochrome C at positive 0.235. In thermodynamic terms, electrons flow spontaneously from NADH to methylene blue, downhill, and then spontaneously from methylene blue to cytochrome C, still downhill. The molecule is not forcing the chain, it is fitting perfectly into the thermodynamic landscape. The result, electrons bypass complex 1 and complex 3, the 2 leakiest sections of the chain, and arrive directly at cytochrome C. ATP synthesis continues. Reactive oxygen species production falls. Methylene blue does not exist in isolation. Its metabolites, Azure B and Azure A, also shuttle. Azure B and Azure A retain the phenothiazinium core. Thionine, the parent compound, has a lower potential, moving it out of the optimal window. Etabanone and MitoQ operate differently at the ubiquinone pool level. Methylene blue satisfies the requirements for ETC shuttling better than any other small molecule identified so far. Join us in Part 2. Visit unbedsidemanners.com for the full story.